Good evening. My name's Dr. Cheryl Forchuk. I'm the Beryl and Richard Ivy Research Chair in Aging, Mental Health, Rehab, and Recovery at Parkwood Institute Research. Now, since this is the second time in a row I've been asked to do this moderation, I'm going to circle back in a minute and give you a commercial as my price. I'm also a distinguished university professor at the Arthur Labatt Family School of Nursing, and a scientist and assistant director at the Lawson Health Research Institute. So now here's my commercial break before we go on, is that now with the Café Scientifique, you get a little bit of a taste of research today. If you find you are not satiated with that taste, tomorrow at Parkwood Institute Research, we have our open house available to the public from 2 to 5. So if you found your way here, you can find your way to Parkwood because it's across the road. Now, I have a bad sense of direction, so I'm probably pointing the wrong way. Which way is it? This way. Okay, this way. But it's right across the road, and we will have a number of interactive displays, puzzles, food, and a lot of opportunity to interact with a number of scientists and their teams in relation to aging, cognitive vitality, mobility issues, and mental health. So back to the main program. My research is in the area of mental health and psychiatric nursing, and I have a particular interest in the relationship between mental illness, poverty, housing, and homelessness, what we often call the social determinants of health. And I'm very happy to be your host and facilitator this evening. And on behalf of the Lawson Research Institute, I invite you to this Café Scientifique experience, the invisible world inside us, exploring the human microbiome. I'd like to extend a special welcome at the front here to our Member of Parliament for London West, Kate Young. So, Kate. So we hope when you get back to Ottawa, you remember how important research is, right? Okay. But good. So congratulations on your recent success as the incumbent for this riding, and we hope you enjoy this evening. For those of you who are not familiar with Lawson, Lawson is one of the leading hospital-based research institutes in Canada. We conduct research that advances scientific knowledge with the ultimate goal of improving health and healthcare for patients. And basically, Lawson is the research arm of all the London hospitals. For London Health Science Centre, St. Joseph's Healthcare, we have one unified research institute. And as academic health organizations, those separate hospitals, it's within their mandate. They have teaching, they have the actual provision of care, but research is actually part of their mandate. And by having one research institute that covers across the hospitals, it means that we really have the ability to follow people and patients across their journey within the healthcare system. And our scientists work within these walls at the hospitals. And that means that the innovation happens right where the care is delivered. And every day, teams of researchers are working very closely with clinicians, patients, and other partners to improve treatments or create entirely new ones. They find innovative methods for delivering services that drive efficiency and reduce costs. And many of the Lawson researchers, like myself and a number of the other people you're going to hear from today, are also themselves healthcare professionals. So it gives us the perspective of really understanding what some of those tough issues in practice are and coming up with some very practical solutions. The Lawson programs include cancer, cardiology, children's health, diabetes, imaging, mental health, neurological disorders, orthopedics, transplantation, and more. And it was recently announced that we here in London, despite the size of the community, actually again ranked 8th in the entire country in the 2019 edition of Canada's top 40 research hospitals listed by Research Infrastructure. And when you really look at that list and see how large the communities are that are ahead of us on the list, I think you'll see what an accomplishment this is for Lawson to be ranked 8th. And to do that, the hospital-based programs, they use several different metrics, including total research income, research intensity, and they look at it by spending per researcher. And so Lawson has received $790 million in research income in 2018, so almost $124 million into this community for health research. That represents a 0.4 growth from the previous year, and Lawson's at the top of the list for research intensity among the large tier institutions, with 612,000 of research spending per researcher. So this ranking has kept London as one of the top 5 hospital-based research hubs in Ontario. So this continued success is the work of hundreds of people across the city dedicated to health research and improving healthcare. And in just a minute, you're going to hear highlights of some of this groundbreaking work and research, specialized research being conducted by researchers, including physicians right here in London. And just as by way of background, in recent years, this concept of the microbiome and the use of probiotics, prebiotics, and fermented food have become very popular. I'm sure probably most of the people in the room have had some of these. We know that there's a strong link between the microbiome, health, and disease, but what does this actually mean? What does that look like in healthcare? What should we do to keep our microbiome healthy? Can we trust all the information out there? Probably not. But we've got people here that can tell you, based on very good research, what we do know. So our talks are really going to tackle some of these difficult issues. And then as well, I hope you noticed with the great spread that was out at the front, we actually have some examples of some fermented foods from local companies. The nuts for cheese, I hope some of you have already tried it. That will be available. It's a gourmet, dairy-free vegan cheese made from organic cashews. I have a lot of vegans in the family, so we use a lot of that. And these artesian products are fermented and aged to perfection. And as well, we have Booch products, locally sourced organic kombucha. And there are bottles on the table and plastic cup for samples. In other words, don't hog it on the table, share it around. So these are 2 of the industry partners of London's Fermented Food project that's taking a rigorous approach to product development and commercialization. Researchers from Lawson support academic support for the project and helping to quantify the health benefits. So this Café Scientifique is different from other scientific lectures you may have attended. It's really meant to engage you in informal conversation. So you think of the idea of a cafe and the idea to sit down and talk, chat about things. So you're going to have the opportunity to discuss, even debate some of these ideas presented by our researchers and others in the audience. So how we're going to do this is first, each speaker is going to give a short talk focusing on what they do and why. We're going to have a quick break, another opportunity to try out that food, and we'll begin the discussion portion of the evening. Now, you all have pads of paper there, and pens. What we really encourage you to do, as you're hearing each of these speakers, if questions, thoughts come to mind, quickly jot them down, because by the time you've heard all 3 people and have that break, you might lose the point, forget exactly what the question was. So do that. Hold on to them. When we get to the panel discussion, you're going to have the opportunity to ask those questions. As well, we have the opportunity to interact with social media, and you see that there. We'll be under the hashtag, as you see, Lawson Cafe, and I encourage you to use that. And moving on now to our first speaker, I'm going to introduce Lawson researcher Dr. Gregor Reid. So Gregor is a fellow of the Royal Society of Canada and a distinguished professor of microbiology and immunology and surgery at Western University. His laboratory at Lawson focuses on the role of beneficial microbes in the health of humans and other life forms. He's produced 29 patents, 550 publications, cited almost 35,000 times, and given 640 talks in 54 countries, including at chapters, church groups, and many lay audience. And I understand he was quoted today in the New York Times. So you've got a great opportunity to hear a very reliable source of information. Gregor has received an honorary doctorate from Aribo, I probably mispronounced that, University, Sweden, a distinguished alumni award from Massey University, New Zealand, a Canadian Society for Microbiologists Career Award, and Western's highest accolade of Distinguished University Professor. So please welcome Gregor to the podium.
Thanks very much. It's a shitty night to come and hear 3 talks about shit, but that's what you're going to hear. I've had a scotch, so I'm good. So the first question is, what's a microbiome? And really, the answer is all of us. But the problem is, we used to just care about this guy in the mustard shirt. Sorry, you wore a mustard shirt. So we used to just care about him because he was salmonella. And if you can imagine in the gut, we forgot about everyone else. And everyone else is the microbiome. And it's the microbes and what you do and why you sit together, not apart, right? So you're sitting next to a daughter or wife or whatever, right? It's not because of chance. And so these organisms, we now realize, are really important. And it's, in fact, I would argue it's much more important than this guy. Sorry, but anyway. So you are, and this is what we look like. Lots of microbes, mostly in the gut, but you also have them in the mouth, and they're pretty much everywhere else. And so you're really made up of a skeleton, tissues that go around it, and microbes. And we've totally forgotten about the last part. We've ignored it in textbooks, and we've forgotten that we really are microbial humans in a microbial planet. So what's a probiotic? You just saw the definition earlier. So it's live microorganisms. You can actually find some people selling dead probiotics. I don't know how you sell a dead probiotic when it's supposed to be alive, but anyway. And it's adequate amounts benefit on the host. So it's not in you unless you've taken them. You know, you hear these people, a dietitian, and says, I have lots of probiotics in me. Well, not if you didn't take any. And it's not kombucha, that's a fermented food. It's not fecal transplant. It's not multiple strains. You go to these health food stores, you've got to buy my products, get 15 strains, and it's billions and billions. Amazing. Never been tested in humans. Did you not get the definition? Hello? All right, so I went to a bookstore. That might shock some of you. I was on holiday. And I didn't buy the books, I took photos of the books because it... Well, you call me a Scotsman? Watch it. So the reason was what they said was garbage, right? And you see this, MD and PhD and New York Times best-selling author, right? Well, in the book, you can see I photographed it, I didn't actually want to buy it because they said that probiotics are fermented vegetables in dark chocolate. What a bunch of crap. Dark chocolate's a probiotic, imagine. And the next guy said they're in you, bugs in you and living in and on you. So there's so much information out there that's totally wrong. And people are buying this stuff. So it is absolute nonsense, and that makes it difficult. So let me try and explain what a probiotic looks like and why you get mixed up. So here's the Uncle Fred. Here's the bifidobacterium. Uncle Fred is in your stomach or in your gut right now. He's a bifidobacterium. Now, this is the probiotic. The probiotic is very, very similar to Uncle Fred, right? But he doesn't have a mustache, right? and he doesn't stay. He passes through. So there's similarities, but this is the probiotic, this is not. That makes sense? Now, everyone will ask me, how do I pick the right probiotic? Here's a website, www.probioticchart.ca. It's a very good starting point. It doesn't have every single product, but what they did was they looked in Canada and said, what's on the market? Has any of it been tested in humans, and what's the level of evidence? So let me give you an example. Here's what it looks like for adult health. Here's all the brand names, the organisms, and this. Of course, at the very back, you can't read it, so I tried to make it a bit bigger. So you go into a shop and they say, you know what? You should buy a line. It's a terrific probiotic. It is if you have IBS. It isn't if you've not, right? So here it is, a line. Here's IBS. And this is what the table shows you. And then you say, well, I'm going to buy Activia. Well, you buy Activia because you tend to get constipation or you get blockage. And Activia, 1 to 3 servings, and it'll help things move along. So that's how you interpret it. And people say, well, what about diarrhea in kids? Are probiotics going to help me? So this is a study that was done, a meta-analysis, which means they looked at all the literature, and they said, let's pull all the literature together and see what happens. And they came up and said, yes, there are some strains of probiotics that can be used for kids with diarrhea. So then the Europeans, it's a society of professionals, they came out and they published saying they recommended certain strains, right? So this is not Mickey Mouse. It's not garbage. You may have heard of necrotizing enterocolitis. So my wife and I had twins. She mostly had them. And they're actually here tonight. And they ended up in ICU because they were early, 32 weeks. So we were in intensive care unit for 5 weeks. And at St. Joe's, and a little baby developed neck. They put a curtain around the baby, and it died. And it was a terrible experience, 1992. And I said, this is a room. The NICU is a room. How do you get beneficial microbes in this room? And the only way you'll get them in is if we bring them in. Because the nurses don't have them. They've got all the drug-resistant bacteria that have been given to the patients and are everywhere else, right? So we have to bring them in. And we went to the literature, and guess what? Everything that is on the left-hand side of this number 1 says, you know what? This can prevent necrotizing enterocolitis if you give probiotics. And it can prevent death in babies if you give probiotics. And so, naturally, I pleaded, please bring them into our NICU. And I had a debate with, at the time, Victor Hahn. And it's on YouTube. It's gone viral, 395 views. Mostly from my mom. But anyway. So finally they did it. Finally they introduced it. 4 years they introduced probiotics to low birth weight. Every kid gets it. Premature low birth weight baby. The rate of potentially fatal neck fallen from around 3% to almost undetectable. Babies aren't dying anymore in London. Why is this not across Canada? Trauma patients. I thought, what's the worst can happen if you get trauma? So let's look. This is polytrauma. In other words, really, really bad trauma. My wife passed today. She called me. She's been a terrible accident. The car's upside down. That's the kind of trauma we're talking about. So they were randomized to receive a symbiotic. So a symbiotic has these organisms, these probiotic strains, plus some fermentable fibers, right? And they gave them it for 15 days in the NICU. This is what happened. The number of deaths significantly reduced, almost halved. Sepsis, more than halved. Chest infections, central line infections, ventilation days. You think, why the hell don't we do that in our ICU? It's only one study. It's from Scandinavia. Maybe we'll catch up one day. Administration of probiotics by mouth can prevent urinary tract infections. This is one of the things I studied for a long time. And we told the people in Holland, don't do this study because postmenopausal women need estrogen. The lactobacilli are not going to work. So they did the study on 252 postmenopausal women. They either gave them Septra, an antibiotic, every day for a year, or they gave them this probiotic that we had worked on for a year. So what happened? Well, the number of symptomatic urinary tract infections went from 7 in the previous year down to 2.8. So that's great. It's more than halved it, and you didn't get it down to 0. Because no one's claiming even antibiotics can get rid of all the infections. What happened to the probiotics? Went from 6.8 to 3.1. Which would you rather take? Pretty obvious. So then we said, what happens if you get an infection? You got to get an antibiotic. But why don't we give a probiotic at the same time? So here's a study. These are 4 studies. This is bacterial vaginosis. We gave metronidazole, which is standard treatment, plus the probiotic. The cure rate was double the probiotic. The cure rate. We did another study. Can't believe it. Oh, guess what? It's just about the same result. 2 different countries. Treatment of yeast vaginitis. Probiotics don't cure yeast vaginitis because it's not a reduction in yeast. Bacterial vaginosis is the loss of lactobacilli. That's not the case in yeast, and yet we still got improvement when you treated with the yeast cream or pills plus the probiotic. What about the brain? Can they influence the brain? Everyone talks about the gut-brain axis nowadays. So I'm going to give you a couple examples. This is Alzheimer's patients treated with milk or 4 probiotic strains for 12 weeks. Only 12 weeks. And there's a mini mental state examination, and it increased in score while the placebo decreased. Now, it's only a small study, but it's suggesting that there's a link between the gut and the brain. And there's a lot of research, unfortunately, an awful lot done in mice, which last time I checked, I don't have a tail. And I don't think a lot of mice work is relevant. So we need to do more human studies, and I think we do way too many animal studies in our country, and they say we can't do things in humans. I think we can do an awful lot more in humans, and we should be doing more in humans, even if it's more expensive. Anyway, here's another one, a randomized, double-blind, placebo-controlled trial, 44 adults. They didn't know what they were getting, right? And this is based on mild to moderate anxiety or depression, and they gave them this probiotic. So 14 of the 22 patients in the probiotic group had reduction in depression scores of 2 points or more on the hospital anxiety and depression scale. In the placebo group, there was no effect, basically. So it didn't affect anxiety, but affected depression. Now, I say to people, this doesn't mean you stop taking the serotonin uptake inhibitors. If you have depression, you need clinical treatment. But maybe if someone is depressed, we can stop them going on to those pills. Or maybe they don't have to have as high a dose. Maybe that's where probiotics can fit in. But again, it's suggesting that there's things going on in your gut that affect your brain. Of course, you know that anyway. You come back after a meal and you're like, ugh, what did I just eat, you know? So there's that brain, gut brain. So prebiotics you may have heard of. What are prebiotics? There will be an exam. You won't get any dessert unless you can repeat this definition. A substrate that's selectively utilized by host microbes conferring a health benefit. What does that mean? It means it's a compound that you cannot digest unless you have microbes inside you. And the good ones digest this substance and then create metabolites that can make you healthier, right? That makes sense? It's not living organisms, it's a substrate. And chicory root, which some of you might know, produces inulin, and inulin, if you take 5 grams of it, will give you a prebiotic effect. And so here, another one of the best ones is human milk oligosaccharides. Breastfeeding. Breastfeeding rates go down in Canada, they should be going up. Highest place in the world for breastfeeding, Rwanda. I thought it was a developing country. What's the effects? I don't have time to go into it. You can just see this is a metabolic health, so overweight and obesity. Here's skin health, allergy, IBD, necrotizing enterocolitis. There's a number of things. So prebiotics have their place. They just haven't had as many studies done on them yet. So another question, should I send a sample of my stool to find out if the microbiome puts me at risk of dementia? I had a guy in Toronto, billionaire. He's all the money in the world, and he says, my parents got dementia. I don't want to do it. Tell me what I have to do. I'll pay whatever. So I said, well, don't send your poop off for a sample. Bloody guy had already done it. So this is what the company said, and this actually turned out the same company. We will maximize your beneficial microbes. We will find out what missing microbes, missing beneficial microbes, missing probiotics. What the heck? They don't even understand. And then we'll tell you what to do. I mean, this is amazing. And so you just, it's simple. You send us your money and we send you a kit. And then you put poop in the mail. How many of you would like to send Donald Trump some of your poop? I mean, what a great idea. Right, so anyway, then you get a score back and it's got this little arrow. Oh, hello there. My arrow says Your health is good. It's your gut intelligence test. The people that do this are lacking in intelligence because we have no friggin' clue. We don't know what microbes are perfect and what ones aren't. And so there's no basis for this. And here's the result. So if you can imagine this is 100%, and the color represents a microbe. So this person has got lots of these blue microbes, and this person has lots of these red microbes. Which one's healthy? One of them's me, and one of them's my student. I don't know. How do you tell if it's healthy based on this? And then they send you this. This is foods for you to eat because of your great profile. Well, here's lettuce. That's a good one. Here's huckleberry. I don't know. I thought that was a farm or something. Huckleberry Finn or character. I don't know. Raspberries. Eat lots of... I mean, it's just total rubbish. And there's companies spinning up all the time on this. I'm not going to talk about this, but I point you to this site, Nature Microbiology Community, because we did a lot of work in Africa. And so I tell this story of a different way to fight HIV, and it's because of this little sachet that makes 100 liters of probiotic bacteria, and 260,000 people in Africa are getting it every week because of this project. But I haven't time to talk about it, so I'm going to talk about fish. Why not? Salmon. This is the West Coast. So this is the global capture fisheries. This is aquaculture. Here's aquaculture for human consumption. Look at it. It's going up. It's going up. Big, big money. What's the motivation? Well, the triploid Chinook salmon, they're sterile. So they're sterile so that if they escape, they can't mate. They don't mature sexually, so that prevents loss of flesh value, but they're very susceptible to disease. So this is a $1 billion industry that is in Canada that we're trying to create, and there's big problems with infection. The death rates are sufficiently serious to answer what can we do. So 50% mortality, huge, huge problem, right? So I stupidly said, well, why don't you give them probiotics? Well, they friggin' went and did it, and it worked. So here's, didn't work brilliantly, but it worked pretty well. So here's the growth rate, the weight gain of the control versus the probiotic. And when you're getting a 20% weight gain, you can see how that translates into more fish surviving, more fish then being able to be sold, and a better industry. So we have a project right now where we're trying to find out why is this, how's it happening, and can we do it? So it's fish. Who would ever imagine I'd ever work in friggin' fish? I tell you, I wouldn't. And then we have all this other project on honeybees, which is also at this nature community site. And the idea was, well, what's killing the honeybee? Well, isn't this an amazing picture? This is taken by Andrew Pittock, one of our students. Amazing picture. So it turns out that bees are in peril, and this is the loss. This is a survey over the years. Here's the percentage. Now, you can take 15% loss in a year of a hive. So you can have 15% of your hives can die. That's fine. It's up to 40%. If it gets any more, you know what? We won't have any food left. That's how important pollinators are. So we again said, what's killing them? Well, a lot of things are killing them. Intensive agriculture, pesticides are two of the big ones. Obviously, deforestation, fertilizers, there's some other reasons, but they're the big ones. And so you can't go to a farmer and say, no, don't use pesticides, because now you're hurting the farmer. So we're saying, well, until we get a means to not use pesticides, and guess what? In Denmark, there's a company that has microbes that you can put on the soil so you don't use pesticides, but that's coming. But anyway, in the meantime, we're killing the bees. So we decided to develop this thing called what we've called a biopatty. It's basically nutrients for the bees, and we put 3 probiotic strains into it, and they love it. They eat it. So my student found out, this is, you can see the low level here, and the reason this is pathogen load. So the worst thing that can happen to a hive is a thing called American foul brood. And it's basically a bacteria that comes in, and it attacks the larvae and the brood, and it's like very sticky. And along comes the Department of Agriculture with a flamethrower, literally, and they burn the whole hive because they don't want this organism, right? And it turns out that we shouldn't really have had to burn the hives that got the probiotics because they were fighting this horrible disease. So my message here is, long may they fly. The take-home messages: think of probiotics as individual products, not a whole category. It's not like, aspirin is good for a headache. Aspirin is a pharmaceutical agent. You wouldn't take aspirin if you had an infection. You'd take another pharmaceutical agent. So think of it that way. Use only strains that have been proven in humans, and if your shopkeeper doesn't know, either don't buy the product or tell them to find out. And the best thing that can happen is the company finds out that, well, actually, we haven't done any human studies. We better do some human studies. That's the best thing that can happen. We should expand the use of beneficial microbes to people across society, including our drug-resistant hospitals, and to our ecosystem, whether it's salmon or whether it's honeybees. These are just two examples. And then lastly, don't waste your money on poop analysis, but you might consider a fecal transplant, which Michael's going to talk about. Because the guy with dementia, if microbes in the gut really do affect the brain, and if somehow we find that the microbes that you have are influencing dementia, then for goodness sake, get a transplant. And I told the guy, I says, you know who you'd want your transplant from? My mother. She's 92, and she can remember everything, everybody. And I'll go and I'll talk to her, and she'll go, Jeannie married in 42, and she lives up the road, and da-da-da. And so the concept is really good, but we need the science behind it, and that's what these guys are going to help you with. So thanks very much.
So thank you. So I think we've got some great insights there, so you can keep your questions on your sheets of paper, and we'll come back to that. And we certainly learned that one size does not fit all when it comes to probiotics, and we need to really know where the evidence is. So our next speaker is another Lawson researcher, Dr. Michael Silverman. And Mike received his medical degree and his residency in internal medicine at the University of Toronto. He carried out his fellowship in infectious diseases at the University of Manitoba and an HIV postdoctoral fellowship at the University of California. So he established the regional clinic for HIV and hepatitis in Whitby, Ontario, and the Durham Regional Tuberculosis Clinic in Ajax. He was consulted on infectious diseases for health organizations in Ontario and was the Director of Infection Control at the Henry Ford Hospital in Detroit, Michigan. So he's extensive research experience and also does humanitarian medical relief in developing countries such as Guyana, Zimbabwe, Zambia, Uganda, and Thailand. He left community practice and returned to academic medical practice in 2014. So he's currently the city's chair chief for infectious diseases and also the medical director of the HIV clinic at St. Joseph's Hospital. He also holds several academic positions with Western University and the University of Toronto. So please, let's welcome Mike to the podium.
Okay, so I'm going to talk about fecal transplant, and hopefully I can convince you at the end that you should care about this crap. So, and I'm going to talk a little bit, so you heard my the intro as to who I am, and none of it had anything to do with poop. So when I went into infectious diseases, I had zero intention to get involved with crap. I was interested in becoming a gastroenterologist, and then I realized how much I hated that smell and decided I was going to go into infectious diseases because I thought pus didn't smell nearly as bad. But around in 2003, I was seeing a patient, and she was about 80 years old, and she had this disease called C. difficile. You may have heard about it. It's an infection that often happens after you've been on antibiotics. The antibiotics wipe out the normal, healthy bacteria in your gut, and this bad bacteria, C. difficile, overgrows. An analogy would be like if you had a store and you had a couple of cats. You never had a problem with mice until somebody came in and killed all your cats. Now you get overrun with mice. So And no matter how many times you try to put mouse traps, you're not going to get rid of all the mice until you bring back a couple of cats that are going to bring them under control and bring them under control long term. So a study came out from Scandinavia where they gave fecal transplants and they put it in through a nasogastric tube, a tube down through the nose into the stomach, and it seemed to work pretty well on a number of people. And just then, you know, as I read that paper, and this woman came in, she'd been in the ICU 3 times. She couldn't afford the medicine anymore. It wasn't covered by the provincial drug plan. She had already put a mortgage on the house for all the costs that she'd put into this medicine, and she was afraid if she ran out of money, she was going to have to go back to the ICU and she might not survive this time. It's important to realize C. difficile, we think of it as just diarrhea. This is really diarrhea from hell. It's got a mortality of 15%. 15% of the time when people get this, they die. When you think a heart attack has a mortality of 5%, this is a serious diarrhea. So people get scared when it happens appropriately, and they get scared when they know it's going to happen again. And many people, it can happen over and over because it's like all the cats are gone. So they put a nasogastric tube and they can get rid of this. Well, by putting poop in. So we said, okay, we'll try to do this in you. And we tried to do it. It was not easy because, you know, where do you do it? And then, going to put a nasogastric tube in. We eventually were able to do it with enemas. We stuck it up the bum, and we did it a couple times. But you may have heard there was a lot of difficulty for the first number of years on, how do you administer it? What's the best way? Where should it be done? You've tried to do it in the emergency department. They say, this isn't an emergency. I don't think we should do it here. You try to do it in radiology, because that's where we can put down the tubes. And they say, well, that's not really, a diagnostic procedure. I don't think it should be here. Well, the OR, they said, that's not clean. We're not doing it here. They said, speak to pharmacy. Pharmacy says, that's not a drug. Like, we're not, and they said, well, get the appropriate permission. From who? Everyone we ask says it's not in their department. Speak to the transplant group. And they said, this is not exactly a kidney. So we don't do those. So eventually, because of these administrative difficulties, we had to shut down and say, we just can't maintain this. So some people came up to me and said, well, we want to do this ourselves because mom or dad or whatever had this happen so many times. We read about this. We saw one of my patients made the mistake of telling the journalist about it, and then ended up on CBC. And I got people coming from all over going, can you give me some poop? So, and they said, well, if you're not going to, you should, please do it. But if you won't do it, then we're going to do it ourselves, because it's not that hard. I mean, we read about how you do it. Basically, you take a blender, you put some poop in there, you put in some salt water that we can get from Shoppers Drug Mart, pour it into an enema bag and squirt. It's not that hard. Like I had described it. It's not that hard, And well, how did we come up with that? Well, we came up with that approach because, we had to figure out how to do it, and I thought, okay, poop is sort of the consistency of really hard ice cream. So how do you make a smoothie? So it's about 4 parts milk and 1 part ice cream, and then you can squirt it, So I went to Shoppers Drug Mart, I think they'll probably get upset if I say that's where we did, but and just got an enema bag and got some sterile saline, and you put it in and squirt. So he said, we're going to do it ourselves. We heard about how easy it is to do. So I said, all right, at least let me screen your family member and make sure they don't have any diseases. They're not like a salmonella carrier or something else. And I can't stop you from doing it in the home. It's hard for me to do it in the hospital because, we have this administrative difficulty. And this was across North America. Hospitals didn't know what to do with this. So a bunch of people started doing it. And this was a funny, so we ended up publishing, 7 out of 7 people who had come in and said, we did it ourselves in our bathroom. And it worked. All of them, they'd had episodes of C. difficile. It had 4 or more episodes. Each time they got really sick, ended up in hospital, and now they did 1 squirt, and it's over. So, and it got widely reported in the lay press. It was all over the place. I couldn't even read. I got, from between Austria and Australia, we got things, and in all kinds of languages. I'd Google myself, and I'd find out in German they're talking about me. Kanadische Gastroenterolog. I am Kanadische. I'm Canadian, but I'm not a gastroenterolog. I'm an infectious disease guy. Do it yourself. And I don't know what else is. I don't know what else they're saying about me, but they're saying so. But it clearly, eventually more and more data came out showing that it works. And it clearly saves lives. And now it's become the standard of practice for C. difficile, for multiple recurrences of C. difficile. For severe cases that are in the ICU, we do it sometimes, but also particularly for recurrent cases. In Canada, Health Canada says it's not experimental, we can do it now. The FDA initially said no, and then they said, okay, but you must make sure that you can do it, but must inform the patient. Like they think we're running up to people when they're not looking from behind. So, I don't know, maybe somebody in the States is doing that, I don't know. But that wasn't generally our approach. So, so what's happening? What's the science behind it? Now, this is a bit of a busy slide, I'm sorry, but so this was our group, and there was another group, and basically what we did was, the thing is the diversity. How diverse is the number of organisms you have? So if you got lots of different bugs in your colon, that's probably good, and that's healthy, because we don't know which ones are the critical ones, as Dr. Reid said. So we don't know, but we know that the more you have, the more broad, the more protected you are against C. diff. And what they found was that, you know, if for people with recurrent C. diff, like in our study, it was low pre-transplant. You have very little diversity, very little different numbers of different organisms. After the transplant, it increased markedly. The donors that had lots of different organisms, not just one dominant one. But before, it's mostly C. diff and these other bugs called proteobacteria. You may have heard of E. coli, these gram-negatives, but they don't really help prevent C. diff. So you got mostly C. diff and E. coli, and then afterwards you got these other bacteria, Firmicutes, Bacteroidetes, anyway, these other organisms that seem to inhibit, they produce what's called short-chain fatty acids, things that are nutrients that help the cells lining your colon live. The bacteria actually produce nutrients that the cells need to live. And when we do the transplant, those show up. So why is that important? Well, it turns out that this may be important in many diseases besides C. diff. So this, medical people, we always call these things cartoons, despite the fact that they're not funny at all. But so this is the lining of the gut. And we got all these bacteria within there. And these bacteria can produce many different byproducts. We used to think they just sit there, they don't do anything, they just watch. But actually, they're very metabolically active. They are producing lots of things. And some of the things it turns out they produce, like TMA, can get turned into your liver and can cause hardening of the arteries. And there's more evidence that atherosclerosis, coronary artery disease, stroke may be related to what bacteria are in your gut. But the other thing they do, they make these short-chain fatty acids that tighten the connections in these cells so things don't leak out. But they also enable you to absorb nutrients. And these short-chain fatty acids actually can be absorbed and give you calories. Some people say, all I do is eat celery and I'm 400 pounds. And we all snicker, going, yeah, I'm sure they eat celery. I'm sure if you looked under their bed, it's all chocolate bars. But it's turning out that some people do have a microbiome that makes them very efficient at absorbing calories. And they really, truly do. And we got to stop snickering. So they can absorb, but also the other thing is that this allows inflammation to occur, because when these toxic products get across, when inflammation can occur, and when that inflammation can occur, it can make you less sensitive to insulin and make you more likely to get diabetes, adult onset diabetes. Even it can make you more likely to get type 1 diabetes, and I'll talk about that because that's actually autoimmune. And it can affect the liver and give you fatty liver, or non-alcoholic steatohepatitis, NASH, which is scarring in the liver, which is becoming the most common cause of cirrhosis in North America right now. How could this cause type 1 diabetes? Type 1 diabetes is when you start producing antibodies and you destroy the insulin-producing cells. So you shouldn't be able to do that. Bacteria should have nothing to do with that. But actually, when these junctions between the cells are loosened, then bacterial products come through. These are what's called, and parts of the immune system, the antigen-presenting cells, the T cells, the B cells, can get irritated and turned on by all these different products that don't belong there. And some of those products can look like other proteins we have, like proteins that are on islet cells, that are on the cells that produce insulin, and make us start attacking our own cells. And by doing that, that can lead to damaging of those cells and causing type 1 diabetes. It seems like one of the central ways that these having, an inappropriate microbiome is by making this stuff leaky, the gut leaky. So the question was, in fact, there's many different diseases that could do this. Could we change that? Could we, if the microbiome, if the way the gut bacteria are affecting us is by making the gut leaky, is there anything we can do to stop that? So we did a study that's just completed where we compared people. These are people who have fatty liver, so they have this, and we think with people with fatty liver usually have an abnormal microbiome. And we checked how leaky their gut is, and the higher the number, if it's above the red line, they have a leaky gut. And then we gave them either their own poop back, an autologous transplant, but nobody knows who had their own. And some of them we gave it from a healthy young person. And if you got it from a healthy young person, your gut got less leaky. And if you got, now randomly, not too many people had leaky, but somebody who did have a leaky gut, it got better, but somebody who didn't have a leaky gut got leaky gut. If you got your own poop back, it didn't seem to make a difference. But if you got somebody else's poop and your microbiome changed, your gut stopped being leaky. So does that matter? Well, the gut microbiome and the leakiness of the gut is thought to be involved in many different diseases. Obesity and metabolic syndrome. That's the syndrome associated with high cholesterol, high blood pressure, central obesity. It seems to be related to a leaky gut. Colon cancer, especially on the right side. Sclerosing cholangitis, scarring of the bile ducts. Inflammatory bowel disease. Crohn's and ulcerative colitis. That one's not as clear, but it seems to be likely. Irritable bowel, and you heard about irritable bowel and anxiety. Autism, because of the gut-brain axis. Depression, anxiety, multiple sclerosis. We just completed one and are about to start a new study with MS. Asthma, rheumatoid arthritis, all these autoimmune diseases, systemic lupus, juvenile rheumatoid. Presence of superbugs in your gut. By getting healthy bacteria, we can get rid of some of those. Cancer, and there's a lot of there's increasing studies, and one particularly out of London that we're doing, that is where it seems that the response to those new somewhat miracle drugs, a big step forward in terms of the immunotherapy. It seems to depend on what's in your gut. And the side effects that are related to them is related to what's on your gut. And we're doing a study with melanoma, about to do another study with renal cancer, and in fact, even HIV. Now, how do we give them? How do we give transplants? In mice, it's easy. You put poop in front of them, they eat it. Dogs, as you can see, will usually drink from the toilet. Humans, most humans will not. So we can do it by enema as one way, colonoscopy where we squirt it up the tube, a nasoduodenal tube, we put a tube down. Capsules. Most people prefer to eat it as a capsule, especially if it's got no smell. And Dr. Parvathi's in the audience, makes the capsules. We're one of the first places in the world to make these capsules. And And then you have to make sure that these bacteria, and you heard from Dr. Reid, some organisms just go through, some organisms stay. If it's C. difficile, it's easy because there's almost no organisms other than the C. difficile and these few other gram-negatives. So whatever you put in is going to stick. But if you've got a lot of other organisms, because the person's got MS or other diseases where the microbiome's off, but not off so radically, then you may have to give an antibiotic before to get rid of the normal bugs so the other ones come in. It's sort of like throwing a couple of seeds into a pine forest. It's not going to take over. But if you kill all the trees first, then you throw in a couple of seeds, then the new stuff will take over. So sometimes we might have to give an antibiotic to get rid of what the person normally has to make the new stuff take over. And then we wash people out with this stuff, polyethylene glycol, just like you're getting a colonoscopy. And then we repeat it. And we've been maintaining a frozen stool bank. People say, that's okay, I got a friend, I got my brother, he's great. We used to do that. And then we find out, how do we discuss the fact that, well, your brother, he's got a problem. he didn't know it. Now we got to explain why, it was when he was in the army, maybe he picked up something. So we don't want to have those discussions. And generally, unfortunately, as you get older, the microbiome becomes less diverse. We have less, so you got to get healthy young people. And a lot of people who have these things are maybe older, particularly with C. difficile, they're in their 80s, so their friend is also in their 80s. And everybody they know is in their 80s, and it's hard for them to find. So we maintain a stool bank. And we actually did a study looking at this. How easy is it to become a stool donor? Because we do more and more and more screening. Well, we screened 46 donors. We took their history because if the microbiome can be associated with obesity, we don't want to take it from someone who's obese because then we're going to be giving the other person obesity. So if it can be associated with depression, we don't want to take it from someone who's got a problem with depression. We do physical exams, we do blood, we check them for everything as if you were giving a blood transfusion, as well as all the diseases that can be associated. But for a blood transfusion, we don't check you for salmonella. But we got to check for all the things that could be in your poop, as well as, people have sex, and sometimes those things can end up in your poop too. So sexually transmitted diseases and multi-drug resistant organisms and all this stuff, we check them for. So only 5 out of 46 passed all the tests. Sometimes because they had these other organisms, Helicobacter, which can cause ulcers, all these things. And then many of them, but we also, and then we refuse anybody who's recently been to the tropics where the water may not be clean, they've had a new sexual partner, and so of those 5, 4 of them ended up like going away on holiday or something. So afterwards, we only had 1 out of 46 was suitable. It's said that it's easier to get into Harvard than it is to become a fecal transplant donor. Your odds are better. So these are very healthy people. And it costs a lot. To find one donor, it costs us $20,200 to be able to do it. And it's not funded by OHIP like we had to. It's a loss. So, but this way, when people come in, they can get treated. Because it's not like this 80-year-old with recurrent diarrhea is going to have 46 people who'll come forward and donate. So we need to maintain a bank. So now there's so many different diseases. It's really the gut microbiome. It's like a new organ. We used to think it didn't do anything, but to me it's like before we used to think the little gland in your neck doesn't do much. It's just a fatty thing, and then we found out it's related to obesity and depression and hair loss. That's not me, but anyway. But and we've said, oh no, it's the thyroid, and it's doing things all over. Well, it's the same thing with poop. We thought it did nothing, and now we're finding it's doing things all over. It's very active. So we're using it for more and more diseases. This is the new prescription pad. And so the question is after this, would you give a crap? And if you're thinking about being a donor, whatever, including a poop donor, then please contact Seema, Dr. Parvathy, because we'll screen you, and hopefully you're one of the lucky 46, and because we're always looking for donors. Thanks very much.
So thank you, Mike. I think we learned a few very interesting things today. In a pinch, how to have a home-done fecal transplant. We know where to get those supplies. It's always good before the Christmas season to have ready excuses for weight gain, so you've also got some of that ready to go. And so thank you. So next to the podium, we have our final speaker of the evening, another Lawson researcher. It's just total coincidence we only have Lawson researchers here. And that is Dr. Jeremy Burton. Jeremy is a graduate, am I going to mispronounce this? Okay, the graduate of the University of Otago. Okay, thank you. In New Zealand. He currently holds the Miriam Burnett Chair in Urological Sciences within the Division of Urology, Department of Surgery, and is cross-appointed to the Department of Microbiology and Immunology at Western University. He's interested in how the microbiome influences urological conditions and what affects the microbiota, such as diet, disease, fecal transplant, treatment, and the use of antibiotics. His entire research career has focused on the relationship of beneficial bacteria to human health. So please welcome Jeremy to the podium.
Thank you very much. So we have an intimate relationship with our microbiota, and this is a slide that shows that. This is Strep salivarius. So if you do that, you've probably got some on your finger now. So who here has got Strep salivarius? If you don't have Strep salivarius, you probably have halitosis. So who's got Strep salivarius? That's what I thought. I didn't show this slide just to poke fun. I showed this slide because these are sitting on the top of a mammalian cell. And when these bacteria sit on the top of the cell, you can see these projections come out to meet the bacteria. And if you look even closer, then the bacteria sends out these little sort of connections to talk to the host. So there's these very intimate relationships going on. This is just one of hundreds of bacterial types that we have in our body, and we have a special relationship with our microbiome. So why is that? It's really because bacteria have always been here. So if we think about the Earth as 4.5 billion years old, 3.5 billion years ago, the first sort of bacteria turned up. No humans. Protists, 2.1 billion years. Plants, animals. And then 7 million years ago, so 1% or less of the time, hominids, not humans, hominids turned up. Humans 120,000 years ago. So we have had bacteria and humans and and all these life forms in the presence of bacteria for such a long period of time. So it's no wonder that we have essentially grown up with bacteria while we have this relationship. There's more microbial cells, more genetic material in our microbiome than our own. So every human system is affected by the microbiome in some way. So if you don't have a microbiome, and people have done mouse studies which show this, you become adversely affected. You don't develop properly, your immune system doesn't develop, your gut doesn't develop properly, and you're stunted. This is a germ-free mouse compared to a normal mouse. So you don't develop the same way. This is an organ, as Dr. Silverman said, in its own right. So at the moment, we're kind of abusing microorganisms a little bit. And this is sort of a diagram to sort of illustrate that. So this is antibiotic prescriptions in the United States. And you can see that these are heavily prescribed areas. This is obesity. And you can see the correlation. Coincidence? Maybe. If you overlap diabetes and a number of other diseases, you will see the same trend. So where we're abusing microbes, we seem to be having more disease. And although antibiotics will change everyone's microbiome differently, we know that it does change it. And we know that in just about every type of disease there is, the microbiome changes, although it was usually unique to that particular person, whether it's inflammatory bowel disease, diabetes, and what drives that particular change. This is Gregor. He's a collaborator of ours, and we worked on a project in China, and he coined this term, ridiculously healthy. And we were studying the microbiome of a big Chinese group. And the reason why we're studying it is because we wanted to try and define what was the healthy microbiota. And really that was a naive thing to do at the time. This study was published a couple of years ago now, but we thought that if we could figure out what the healthy microbiota was, maybe we could potentially drive people towards it. So we did a study in China, and we looked at 1,000 ridiculously healthy people. And they're ridiculously healthy because they never smoked, never drank, never took antibiotics, were never unwell. And although the slide hasn't turned out particularly well, we had a range of people from 3 years of age to over 100, including 200 people that were over 96 years of age. And what we found was that the diversity of these super healthy people was pretty much the same as they were when they were 30 years old. So either your microbiome is helping with you being healthy, or it's a good indicator that you're healthy. So this was a really interesting finding. So if we want to get a healthy microbiome, how do we do that? Well, there's not a lot of ways that we can actually do that at present. We've been really good at killing bacteria and microbiota, so antibiotics and that sort of thing, but really we're not very good at promoting or supporting the microbiome or replacing, as you've heard about fecal transplant as a particular option. But really that's a kind of a crude process, and maybe we can do better in the future. So this started off a little bit with this guy here. This is Elie Metchnikoff, and he's known as the father of probiotics. But he actually got his Nobel Prize for discovering phagocytosis, which is an immunological phenomena. But he is known as the father of probiotics. But what probably isn't known about Elie is that he was a total germaphobe. So we give him this great accolade of loving bacteria, but actually he hated bacteria. He did, he hated them. So he thought that the gastrointestinal tract was basically a sewer waiting to cause disease in our body. He was terrified. He made people peel his fruit and vegetables from. He would only eat cooked food generally. So he actually hated bacteria, but then he saw Bulgarian peasants eating probiotics, and he thought, well, maybe if you took good bacteria, you could overcome this. And so probiotics was born. So people have a lot of naive concepts about probiotics. Probiotics, people think, we have this decimated microbiome, we take probiotics and we restore it. This is not the case. And you'll see a lot of claims on products and in the media and other things about, restoring natural balance, replacing, recolonizing, supplementing the diversity. This is just not true. This is what really happens. Probiotics go in and probiotics go out, and it looks exactly the same. They don't stick around, they keep moving. That doesn't mean that they don't work, though. That doesn't mean that you don't see a clinical effect. This is a study that we did in rats that we collaborated on, and in some of these groups, so this is a microbiome analysis, and I won't go into it, but essentially there's lots of groups here, and some got probiotics and some didn't. Some got huge doses of probiotics, and we didn't change the microbiome one little bit. So they went in and they went out. But the difference is that those that got probiotics, these animals didn't go on to heart failure, so they actually did work. They were doing something. So that doesn't mean probiotics don't work. They just don't stick around, but they do something. And as Gregor alluded to, there's good information available to find out what probiotics can do what. They're not miracle things. They won't cure everything, and they shouldn't be expected to be put into that kind of category. a question that I get asked being in the field is, should I take a probiotic every day? Should we eat bacteria every day? And the answer, I think, is yes. I don't have a really good reason, but if you look at some of the information around us, I think you would agree. So this is Drosophila fly. We use this as a model quite often in the lab. And if you starve it of acquiring new bacteria, they die quicker. If you treat them with antibiotics, they die quicker. So they need bacteria to survive, and they need new bacteria to survive constantly. Now, we don't know if that's the case for humans. You know, we're far more complicated. It's not easy to actually determine this. But I can tell you that our diet now, if you're eating a typical North American diet, is actually kind of pitiful when it comes to the numbers of bacteria in it, 1.4 times 10 to the 6. So 1 million bugs, which sounds like a lot, but actually in terms of microbes, it's very, very small. It equals to about a spoonful of yogurt or maybe one kiss from your grandma, maybe. Not a big sloppy kiss, that's for sure. Our microbiomes have changed. They're not the same as they used to be. So they dig people up from ice. This morning I saw they dug up a prehistoric puppy, but they're digging up people from ice and from some from not long ago and some more distantly, and they've looked at their microbiome. And their microbiome is quite different to ours. So we know that it's definitely changed. And we also know that if we compare our microbiome in urban areas to people that are still hunter-gatherers, and this is Jeff Leach, who's an anthropologist who went to Tanzania and lived with the Hadza hunter-gatherer tribe, that first of all, his microbiome was different, but if he lived with them, it changed. So that was quite interesting. And my colleague Gregor was involved in the study because Jeff collected a number of samples. And the interesting thing was that, okay, not only is the microbiome different, but it changes with the seasons. So, you know, these people collect different things at different times of year, whether it's wet or dry, and the microbiome changes, and also they're acquiring new bacteria. We don't do that anymore, not at all. And the reason for that is that we now guarantee our food source, which has proven useful for some things, but maybe it's come at a cost. You know, now we don't pasteurize milk, and we have other food safety and hygiene practices which have extended, you know, the number of people that survive, and, you know, obviously saved a lot of lives. But maybe during some of these processes, during agricultural revolution, farming practices, antibiotics, chemicals, maybe we have lost something that's important. And so maybe we should try to think about getting some of that back. So how do we start to get that? We could all move to Tanzania, but I don't think that's very practical. So one thing that we thought that is missing is fermented food, because every culture has a history of fermented food. So originally, fermented food was really for preservation, because you'd get a lot of food at one time of year, you'd try and store it, and preservation was a great way to do that. Later on, we started producing useful things, acids and alcohols, and then, you know, obviously it tastes interesting, but now maybe we should be thinking of it for health reasons. So every culture on this planet has a history of fermenting food. Doesn't matter if you have a dairy background or a fish background or eat meat. Every culture, and this is just a small list, has a history of fermenting foods to preserve food. But we don't do that as much anymore. Even in Canada, you know, this was fairly common practice. So maybe we should be looking at, you know, giving fermented food back. Maybe this could add something to our diet and health. So the problem is that these bacteria aren't necessarily the ones that we're missing in our guts, but perhaps that we can add them in and get some health benefits like we do and what we see with probiotics. So we started working in the fermented food area just a little bit with a couple of local companies, and you'll see some of their products here tonight, just to work out what's in them, because fermented foods can be a very rich source of bacteria. And you can see in this chart here, we've got, 10 to the 10, which is a huge amount of bacteria in some of these products, whereas down before we were down here before in the diet that we currently have. So we can potentially get a huge number of bacteria back into our diet. So you'll see the Booch product here, and I don't take any money from Booch. I just drink their stuff when they give me free stuff. So it's very good. I enjoy it. It is loaded with bacteria, different bacterial types. Each colour here is a different bacterial type. It's tasty. I like it. As per nuts for cheese, it obviously also contains a whole range of different bacterial types. The interesting thing about the nuts for cheese product that we found is that the more you ferment it, the more that the cashew allergen content drops in it. So actually, if you're mildly allergic to cashew, maybe by the time it's fermented, you could potentially use that product. So interesting other findings too. So in the future, we might try to find the bacteria responsible for that and to make products that will reduce cashew allergen for those sorts of people. So what about using fermented food maybe to treat and prevent disease? So we have a big interest in kidney stones, and actually fermented food is quite the sideline for us. So kidney stones are increasing dramatically in the West. They have a large recurrent rate, and they actually also have bacterial links. So this is a kidney stone in a CT. You can see that here. And although it's not quite as big an operation as it used to be, it's now a keyhole surgery. It's still a very costly operation, especially if you're going to have to repeat it every 5 years when you get a recurrence to get these stones out. So there is a bacterial connection to kidney stones. You might think that that's a little bit strange, but if you're lacking certain bacteria that can break down oxalate, and oxalate is a big component of the most common kidney stone, you're more likely to get kidney stones. And also, the latest data suggests in a very big study from the United Kingdom that if you're exposed to antibiotics, that your risk skyrockets, especially for the drugs the antibiotics that they use to treat UTI. So that's another interesting finding. So just to explain how we handle oxalate, which is a large part of kidney stones, we take in a little bit of oxalate in our diet. So it's in green leafy vegetables, tea. So you'd think that vegetarians and vegans would be highly susceptible to kidney stones. Well, they don't get kidney stones as frequently, which is interesting. So we take in about 20% of our oxalate from diet. But actually, by and large, most of it comes from metabolism. And it's a side effect of metabolism and will go out in kidneys. But actually now what we think happens is that not only it goes out through the kidney, but actually some of it gets sunk into the colon. And the bacteria in the colon utilize it and actually draw it across. So if you're using antibiotics, some of these bacteria are obviously going to be knocked out, which isn't a good thing. So we use a model, they didn't want to be identified, where we give kidney stones to this particular model. For some reason, ever since they've started working in the lab, they've gotten kidney stones, and it greatly affects them, but there's not actually a very good model for kidney stones other than humans. And these particular models, they don't have kidneys, but they have a nephron-like thing called a malpighium tubule. And we can give them a diet and we can get these calcium oxalate crystals forming. So we can generate something that's similar to a kidney stone. And they can also poop it out. And we can measure the amount of crystals that come out in the poop. So we can test different treatments with this particular model. And we can see actually how quickly they die or don't die with our particular treatments. And actually we can also even live image Drosophila as well to see what's going on. You can see that there's the main bit of the kidney, well, not a kidney stone, but in the malpighium tubes where the crystals are formed. So we've taken some of the fermented food bacterium, Bacillus subtilis is one, and this is an organism which is widely used in Africa and Asia to ferment soy and other things. And this microorganism loves oxalate, loves to eat it and can utilize it. And when we give it to our model, we actually find that it reduces the amount of crystals that form. You can see this here. So the flies that have got the bacteria reduces the fluorescent of those crystals that you would usually see. And also they feel a lot better because they're crawling around, they're performing tasks a lot better. So maybe one day in the future, we'll be using fermented foods for more medically orientated purposes. So I've quickly gone through a few things today, but I've got 3 messages for you to take home. First of all, bacteria are important. We have grown up with them and we need to understand them better and their role. So our microbiota is also different from our ancestors, which have evolved over time to a certain set of bacteria. So we're in a new sort of era, essentially. And lastly, maybe consuming and supporting the bacteria that we have are going to be important for health outcomes. So I'll leave it there. So thanks very much.